Thank you very much for inviting me. It's a pleasure to talk to you today. And I chose this title partly because there's a lot of excitement about RNA technology, but there's also quite a lot of controversy. And I think it's good to look at both of those issues. So just starting at the kind of early stages, just thinking about how we define what a vaccine is, because there are many different types of vaccines. It's usually a biological preparation that provides active acquired immunity to particular infections or malignant diseases. And that obviously was developed initially from Jenner a long time ago, when he worked out that by vaccinating using material from the cowpox virus, he could induce immunity against smallpox. And I haven't included it here, but even then there was lots of anti-vax sentiments at the time, and people were worried that by being vaccinated, they might start to grow things like cow parts. There's lots of beautiful presentations of some of those in pamphlets and newspapers at the time. And then moving to relatively current times, the COVID pandemic, where most of us had vaccines, and I suspect a great many of you had RNA-based vaccines and may well be continuing to if you're getting vaccine boosters. Now, RNA technology didn't suddenly arise overnight. And I think, it scared some people because they felt that the vaccines had been developed too quickly and the technology was too new, and therefore it was somehow dangerous. But it's built on a huge amount of work, starting from the early 60s, when RNA was discovered, understanding some of the ways RNA works, and then into the 80s when people started to be able to synthesize RNA in the test tube, which was a real breakthrough. And then learnt that essentially RNA is extremely fragile. It breaks down very easily. And so they worked out ways to stabilize it, deliver it into cells. And then the first clinical trials came through in the early 20,000s. And initially, they weren't a fantastic success. So it didn't mean that they were unsafe, but Moderna, CureVac, and BioNTech had tried some vaccine targets against things like influenza and rabies early on. And although they gave immunity, the level of immunity wasn't as good at that stage, wasn't as good as they were hoping for. And so they were reiterating their approach, their technology, and were really poised to bring it into large-scale clinical trials when the pandemic hit. And because these companies didn't have a licensed product, they were very, very motivated to move as fast as possible. And the other thing represented in this slide is Drew Weissman and Kati Kikiro, who won the Nobel Prize for medicine in 2023, because they worked out that you could include a modified, what we call nucleoside, so one of the building blocks of RNA, but which was slightly different chemically, although it's still a natural product in terms of it still occurs in humans naturally. But this was actually found to stabilize the RNA and reduce the response to RNA that all of us make when we see RNA that's introduced to a cell from outside, because often your innate system recognizes this more like a virus rather than natural RNA. And this type of modification reduces that recognition. Now, in terms of the genetic flow of information, obviously RNA is the middleman, if you like. So DNA that makes up your chromosomes it stores all the information for every protein you may need to make during your lifespan. That library, if you like, can be modified by epigenetic modification. So some genes can be turned off or turned on, but essentially that doesn't change. Or if it does change, it may change because of environmental influence that may cause a gene defect or trigger the development of cancer. RNA essentially is produced in the nucleus when the right circumstances are there to start to say we want to make this protein. And so RNA polymerase will make the messenger RNA, and that then comes from the nucleus into the cytoplasm and engages with the ribosomes, which will translate that genetic code into protein. It essentially assembles the protein one amino acid at a time, based on the triplet codons that encode for the different amino acids. And the reason that RNA has a very short half-life is that provides a mechanism for being able to control the amount of protein that you make. So if you want to just make protein for a very short period of time, you'll have a short burst of messenger RNA. It will make that protein and then it will break down, and there'll be no more message to feed to the ribosomes for that particular protein. If you need to make that protein all the time, for things like housekeeping genes that need to be produced to keep the cell alive, that RNA will be continually produced and will continually engage with the ribosomes so that you make that protein for a prolonged period of time, or however you want. So this provides some kind of advantage and also a disadvantage when thinking about using RNA as a therapeutic. It means that it cannot change your chromosomal material because it doesn't enter into the nucleus, and it doesn't hang around for a long period of time. So it makes no permanent changes of itself. Well, that goes against some of the myths that are out there, because there are many people that have been suggesting that mRNA might change your DNA and also might cause cancer. But essentially, there's no risk of RNA changing your DNA. It doesn't enter the nucleus, and the RNA is synthetically made and has a very short half-life. So we know that these things are essentially not possible, despite a lot of media attention and false information that is out there. And we continue to see information that is often manipulated for those that want to suggest that the technology is bad. The most recent example I can give you one was a review article that I had to campaign for a year to be withdrawn, because it essentially quoted another paper saying this paper demonstrates that RNA vaccines cause cancer. And they were essentially quoting a paper where they were looking at an animal model of cancer. The animals were injected with cancerous cells to induce cancer, and they were using different type of RNA vaccines to prevent the cancer. And one of those RNA vaccines didn't actually prevent cancer, but it's not the same, obviously, as the RNA vaccine being injected into a mouse that hasn't been injected with tumor cells and actively causing cancer. Many other examples. So how do they work? Well, RNA gives us a lot of things that we can use in terms of designing the way it behaves. So in this little cartoon here, you can see the major block, the coding sequence, is what provides the code for the particular protein that you want the cell to make. But the parts at the front end and the back end can be used to influence the amount of protein that's made by influencing the stability of the RNA and the ability for it to bind to ribosomes that then translate that coding sequence into a protein. In order to be able to deliver it into cells, because it is so labile, if you just use a naked RNA and inject it into a muscle, it would be degraded within minutes. So in order to allow you to deliver it safely into the cytoplasm, it has to be packaged in what we call lipid nanoparticles. And these are very beautiful structures. They self-assemble, typically using 4 different types of lipids. One of these lipids is a charged lipid that binds and condenses the RNA into the center of this lipid nanoparticle. And they're about the size of a virus. And because of that small size and their surface properties, they can bind to the outside of cells. They're then endocytosed. And as they get into the cell, they can cause endosomal escape, and the RNA is delivered into the cytoplasm. So rather than the RNA being made in the nucleus, this synthetic RNA is delivered into the cytoplasm. The cell then will see it, it will engage with the ribosome machinery, and it will start to make protein. So it's hijacking a normal process. Now, the really nice thing about RNA, having worked on many other vaccine modalities over my career, is that this is a synthetic process. So we no longer have to make it with animal cells, with viral particles, or with bacteria. So it's actually probably a much, well, not probably, it is a much cleaner process, much less likely to come down with contamination. And it's very easy to quality control because there are only a few components that you have to check in terms of their quality and that they're all assembled in the right way that they will work in vivo. They're also all readily biodegradable, so they don't hang about once they have been introduced into the body. Now, this is a slightly different diagram, but showing what happens. And I want you to concentrate, first of all, on the left-hand side, where it says mRNA. You can see at the top of the slide, here is a depiction of the mRNA sequence that's being delivered. And this might encode, for example, the spike glycoprotein of the COVID virus. It's encapsulated in one of these lipid nanoparticles. It gets into the endosome. It's released into the cytoplasm. And these green and blue structures are supposed to represent the ribosomes. They recognize that and will produce the protein of interest. Now, when we're designing it as a vaccine, we either want it to be expressed so that the immune system can see it and induce antibodies, or be degraded to induce cellular responses following its presentation in association with MHC antigens that talk to your T cells, either class 1 or class 2 MHC antigens. A newer type of technology, but similarly, very similar in terms of it's still just an RNA sequence that we are working on very actively at Imperial, is using what we call self-amplifying RNA. This is shown on the right-hand side. And this carries with it the proteins that are required to copy your gene of interest. And the advantage here is that once it gets into the cell, it makes multiple copies of the RNA that it's carrying, so you get more protein expression, higher expression in association with your MHC for T cell responses, and it's expressed for a longer period of time. So for some indications, we might want a longer level of expression to either induce a more robust immune response, or if we were trying to express a therapeutic, for example, for protein replacement, we might want to move from something with mRNA that might require weekly dosing to something that might require monthly dosing. And just to illustrate the difference between these two technologies, here you can see it modeled in a mouse experiment where the mice have been injected with a RNA encoding for an enzyme called firefly luciferase. And once that enzyme is expressed, we can give them the substrate, and essentially they glow in a way that they can be imaged under a camera. And you can see for mRNA, shown in blue on the graph, you get a very quick expression and then it goes away. And for the self-amplifying RNA, shown in red, it goes out for a considerable period of time. And that shows that with different ways of using RNA, we can start to tailor the level and the duration of expression. Now, one of the advantages of RNA technology is that it is much simpler to go from identifying a gene sequence to manufacturing a product, because it is all based on genetic sequence. The genetic sequence is used to produce the RNA by in vitro transcription from a DNA template. It's purified, it's formulated, and then you get the final product. It allows you to do this at a much faster speed, but also with a much smaller footprint. So when people are making recombinant proteins or going back to making viruses that they then need to inactivate, typically they would use reaction vessels from several thousand to maybe hundreds of thousands of liters. Whereas for RNA technology, this can be done in the 10 to 100 liter volume, so much smaller. It also means that the technology is what we call a platform-based technology. Because if you're making a vaccine against COVID, you're using the same technology if you then say, I want to switch and make a vaccine against influenza. It's still RNA sequence. It's still, by and large, the same formulation and the same process. So it gives a lot of flexibility in terms of manufacturing, speed, and production. Now, this is an example of the genetic code for the Pfizer vaccine. This is the original Wuhan strain. And the parts of the sequence highlighted in turquoise are not encoded. They are the bits at the front and the end of the RNA that tailor the duration and efficiency of expression. But you can see it's a genetic code. It's very simple. And for students in the laboratory, we often say, well, here you are, go and make a COVID vaccine. They can take a DNA template with that sequence. They put in the enzymes, the building blocks, the nucleotides, and they produce RNA that can then be used in the lab for experiments very easily. Now, this has been industrialized, and the sequence is not secret. I haven't stolen it, but it shows you that actually that genetic code is pretty accessible. And we know from current modeling that the mRNA vaccines are thought to have prevented about 2.5 million deaths globally, and about 1.5 in Europe, because it was used probably more in Europe than in the US. And we also now have safety data, real-time safety data, used with cutting-edge technology from over 6 billion individuals, or 6 billion doses of vaccine that have been administered. So we know more about the safety of this technology probably than any other vaccine because it's been assessed over such a short period of time, but using the same type of assessment rather than going back and looking at multiple historical studies to come up with the same conclusions. Now, all vaccines come with some side effects, and most of the side effects for mRNA vaccines shown here are quite often associated with other vaccines. There can be slight differences in the reactogenicity, and we still think currently that the mRNA vaccines can be improved to make them a little bit less reactogenic. I suspect if we did a poll, a number of you would have felt a bit rough for 24 hours post having an RNA vaccine. And we would like to, in the future, be able to dial that out so that everybody has no feeling of either fatigue or lethargy post-vaccine administration. Now, how do they work? When they're injected into your muscle, this is shown a kind of on a different scale. They will be expressed in the muscle tissue, but also some of that material gets to the draining lymph nodes. Here shown injection into the deltoid muscle and the draining lymph nodes that are closely associated. They will express the protein, and that will work to induce a potent immune response. Now, we've been using that technology ourselves to build a number of vaccine candidates. So we worked quite actively on SARS-CoV-2, the cause of COVID, and moved that ahead. But now we're working on new indications, and we currently have a clinical trial funded by the UK Research Councils, looking at a combined vaccine against Ebola, Marburg, and Lassa fever, which are the causes of hemorrhagic fever. And this is because there's a lot of interest in developing vaccines against viruses with pandemic potential. But some of you may notice that we are also working on bacterial targets. So RNA technology can be used against bacterial targets. Here for us, we've been working on Yersinia pestis, the cause of plague. Somebody died in the US in the last few months of plague. So it's not something that's disappeared, although it is very treatable with antibiotics. But people are still worried that this could be used, for example, as a bioterrorist weapon or as a potential threat on the battlefield. We're also starting to look at whether we can use this technology against antimicrobial resistant organisms. And Acinetobacter baumannii is a cause of a lot of antibiotic resistant infections in low and middle income countries. Just to give you an example of the Ebola, Marburg, and Lassa vaccine that we've been developing, you can see here we've already evaluated it in animal models to show that it can essentially prevent death in these animal models very readily. And so we're interested in seeing whether it can give the levels of immunity that we would anticipate might be protective for humans. Now, people are also very interested in expanding this technology to other situations. So I've talked about infectious diseases, but it's also a technology that's being very readily evaluated for use against cancer. And when we think about cancer, again, what we're trying to do is wake up the immune system to recognize cancerous cells. And this can be against what we call common antigens, common cancer antigens that might be shared for a class of tumor. So it might be shared, for example, for prostate cancer or shared for breast cancer. And so you may be able to make a cancer that's used for a number of individuals, but increasingly people are trying to also use it to tailor vaccines against personalized antigens so that in the future, at some point, the promise is that you might be able to go in, have your cancer gene sequenced, and then develop a vaccine that's tailored specifically for the particular type of cancer that you might have as an individual. It's very similar in terms of the approach. You introduce the cancer immunogens encoded in the RNA sequence. They are then expressed by the ribosomes. There are some tricks we can play, again, to try and elicit and wake up the immune response to induce both protective antibodies and T cell responses that can go out and target tumors. Now, in some ways, and this is more general to vaccines as an approach and not just RNA, one of the problems we find at the moment is that vaccines in some ways have been too successful because people are no longer seeing the diseases that they cause. And a typical example here would be measles. The measles vaccine is extremely good at preventing infection, but we know that we need to have a level of coverage to prevent outbreaks that are greater than 90%. And so what we typically see, and we're seeing in the US at the moment, is as soon as that coverage starts to dip below that threshold, we see measles cases coming back. And in the US at the moment, there's been over 1,000 recent cases of measles, whereas in previous years, it was very well controlled. So we sit in this situation where we tip between people thinking, oh, it's a good thing to have vaccines, where they see these outbreaks, to going back to wondering whether they need to have the vaccines because they've no longer got used to seeing the disease around. We're also finding that it's becoming increasingly expensive to develop new vaccines. And part of the reason for that is that some of the more easy vaccines have been developed, and more challenging vaccines are increasingly requiring more research and more money. So what do I mean by that? Well, vaccines that have been hard to develop would be effective vaccines against HIV or dengue fever, vaccines that with improved efficacy against malaria or TB, are really things that we would like to have, but are very high risk endeavors. And so most vaccines cost at least $1 billion to make. And the higher the risk, the less likely that pharmaceutical companies will invest those dollars to get to the end game of developing an effective vaccine. Now, RNA technology, again, can help in this area because it can at least get us into clinical studies early and get an understanding as to whether these vaccines induce the type of immune response either for oncology indications or in terms of, for example, perhaps seeing whether we can induce broadly neutralizing antibodies against a target like HIV in a safe and effective way. So what we see in terms of public acceptance, we tend to flip from people thinking it's a miracle, it's fantastic, it works well. And that certainly was the case for a large number of people during the COVID pandemic, to then feeling mistrustful and wondering whether they need to keep using vaccines. So there are some people at either end of the spectrum, those who refuse all vaccines and really aren't open to rational discussion, to those who are happy to accept all of them. But there are many in the middle who are often unsure about what they should accept and what they shouldn't accept, and many individuals who are not sure if it's right for them as an individual. Beyond that, obviously, there's also issues around access and issues around being able to get to the clinic in busy working lives and busy personal lives, that may also make vaccine acceptance more difficult for some individuals over another. Well, one of the things that we've been trying to think about in Imperial and that we are in the process of developing is a new centre where we bring vaccine scientists and allied scientific disciplines to start to think about the future of the way we may use vaccines. So we are establishing what's called a Centre for Precision Vaccinology. And really, why do we need to think about this? Increasingly, we believe that we're moving into an age where rather than making one vaccine that works for everybody, but not necessarily as well for all different individuals, to tailoring vaccines for specific health needs. So whether that's the extremes of age, the very young, the very old, and I think many of us are invested in thinking about the needs of older age, where essentially your immune system is not working as efficiently, and whether we can start to think of ways to induce better immune responses in those with waning immunity as we progress in age. Also, we know there are big differences in terms of genetics, gender, environment, and all of those issues that we would like to start to use this new technology to allow us to get more precision in terms of developing vaccines that are more appropriate for different populations. So how are we thinking about doing this? Well, this is bringing different disciplines at Imperial together to bear on this problem. One of the first parts of that model is to think about the developments in rational vaccine design approaches. And here we live in an age where whether it's a virus or a bacteria, we can pull down sequences from all circulating strains. We can use that to design proteins using artificial intelligence and different modeling tools that now allow us to design proteins in silico and then test them, first of all, in vitro, but ultimately to test them for immunogenicity. And so the acceleration in terms of the design of new vaccine targets and new therapeutics is moving extremely rapidly. We also are bringing approaches to get things faster into the clinical space. And here we at Imperial have been putting together a clinical production facility for RNA technology that will allow us to test early designs to see if they induce the type of immune response that might merit them going into larger scale clinical development. This de-risks the huge amounts of money that are often required to move through the development process and scale up that's required ultimately to move to licensure of a product. So one of the examples here is thinking about whether we can define the type of protective immune response ahead of a potential outbreak. And one of those outbreaks that we are already thinking about is avian influenza, which has been circulating most recently in the US. And we know that from a modeling point of view, we're only 2 amino acids away from avian influenza being transmissible from human to human. So rather than waiting for that to happen, we are taking the hemagglutinin protein, which is essential for the virus to bind and fuse with human cells, engineer in those 2 mutations just to the spike protein, not to the virus itself. So we're not creating a virus. And the protein is also stapled together so that it actually can't fuse. It's representing the spike before the fusion process. And we're looking to take that into a very small clinical study pull out monoclonals from the individuals that are vaccinated so we can very accurately map the immune response and predict what would happen if this type of protein was developed by the virus, and understand the type of immunity that we might need to induce, so that rather than waiting for it to happen, we're already prepared and testing potential vaccine designs. And this slide, which I won't go into great detail, is just there to emphasize that now, again, in terms of technology, we can isolate very efficiently monoclonal antibodies from people that have been vaccinated or people that may have seen different viruses. And this allows us both to use those as tools to develop better vaccines, but also to develop those monoclonals as a therapeutic that can be given to individuals that wouldn't respond to a vaccine. So those who might be suffering from immunosuppression or going through the types of therapy that might be required for cancer treatment at the moment that, again, might impair their immune system. So in some ways, it's a win-win situation. We're evaluating potential vaccine immunogens, and we're also being able to identify sequences from monoclonal antibodies. And then the other aspect that we can bring to bear in this new center is the ability to do human-controlled infections with respiratory viruses. And this is work led by Professor Chris Chu at Imperial. And it builds on a history of looking at human infection studies right back from the early 19th century, looking at influenza and common cold research. So we're not reinventing the wheel. We're building on an established process that has been in place for a long time. So it allows us then to look at natural infection in a very controlled manner in healthy individuals and start to understand the correlates of protection. Ultimately, in the future, it will also allow us to look at vaccination and challenge to see if vaccination will prevent infection. And this has already been done for a different virus, respiratory syncytial virus, in collaboration with different companies. And you can see here on the left-hand side, shown in the orange line, individuals who are vaccinated in terms of viral load post-infectious challenge, or those given placebo. And so in this particular study, it was possible to demonstrate that this vaccine was very good at preventing infection. And then the final part that we need to bring to bear is all the new technology that we have from artificial intelligence and machine learning, where we can feed into clinical trials, either trials we're doing ourselves or by looking at trials and data published across the globe, at how age, health, gender, genetics, and environment will impact onto the immune responses that can then be used to better tailor the design of the vaccine and the approaches. So we hope that this new centre will start to give us the opportunities to determine the best type of vaccines that will work on an individual basis. That doesn't mean that we're always making an individual vaccine for every single person, but it does mean that we can start to use vaccines with more precision for different target populations. So pulling that together, it requires to have a good team of directors. And here are my fellow colleagues who bring different aspects to vaccine development. But underneath all of these individuals and working together with them are over 100 researchers at Imperial who are bringing synergistic and allied technologies to play. So we hope that we will start to get this established. We're putting it together by the end of the year. And this really helps us to be future-proof, to thinking how we can start to join up the change in genomic medicine, where more and more individuals will start to have their genomes sequenced. And that's another massive change in terms of the way we do medicine, in that the ability to do whole genome sequencing, the first whole genome that was sequenced cost $100 million. And now whole genome sequencing can be done for less than $1,000. And I suspect it will become cheaper and cheaper. And we may see at some point that most individuals actually have had their genome sequenced and can be used in the design of new genetic medicines. And this brings me back really to the mRNA technology that we can then bring to bear against a range of different therapeutic indications. So these things are not there now, but we're trying to work on them to maximize how we can use these new approaches to make new medicines and get the most out of this promising technology. And I'll stop there and very happy to take any questions.
Yes, very good. Thank you very much, Robin. We have 3 questions. Firstly, from, where are we? The screen's going all over the place. From DR, 2 questions in one. How do you assess the vaccines produced in response to the SARS-CoV-2 pandemic when they have failed to achieve herd immunity so far? And secondly to that, and what is your perspective on the latest development of mucosal vaccines when such immunity is short-lived? Over to you.
Those are great questions. So first of all, I think that there's often a confusion about how these vaccines are working. So they are working extremely well at preventing people developing serious disease. They are limited in terms of their protection against symptomatic infection. So they provide protection against symptomatic infection for a period of 6 months to 12 months, but that's also dependent on whether the virus has made changes so that it can get around the immunity that has been induced. So they are extremely effective at preventing serious disease and at preventing people ending up in hospital. Now I'm blanking on the second part of the question.
It's the
mucosal immunity. So we would definitely prefer to have vaccines that induce mucosal immunity because that would be more effective at preventing infection if you can neutralize the virus before it hits your respiratory cells, your epithelial cells. People are starting to look at how this technology and other technology can be used as, for example, an intranasal vaccine. Still early days, but I think we will see some breakthroughs in that in the coming years. It is a technology that we're starting to look at in collaboration, hopefully with a pharma partner.
Right, thank you. DR then goes on to ask, do you distinguish between the term vaccine and therapeutic in your vaccinology work? The answer is yes there, but
yeah, I think the answer is probably not clearly, because a vaccine can be used in a therapeutic context. So if we're talking about a cancer vaccine, it would be a therapeutic vaccine. If we're talking about a rabies vaccine being used after somebody's been exposed to rabies, that would be being used therapeutically. The technology itself, RNA technology, obviously can be used in a non-vaccine context where it would be a therapeutic. So I suppose whether it's accurate, I think of vaccines as being under that same category. Although you can split hairs and say a prophylactic vaccine maybe isn't absolutely under that definition.
Yes, I see. Thank you. And then he goes on to ask, or she, I don't know who, she's saying, can mRNA technology be leveraged to enhance cell-based innate immunity in the production of natural killer vaccines, or is it confined to aspects of adaptive immunity?
That's a really exciting question, and actually, there's an initiative that's just being launched by a new funding organization, which has put out a call, and they're going to invest, I think, 46 million pounds on asking whether there is technology that can train the innate immune response to be better able to deal with infections. So, is it possible to encode strategies that may then allow you to have a better level of innate immunity that may not be adaptive, but might be effective if it was pre-existing to either prevent or reduce infections?
Thank you. Thank you. I had one myself earlier, which I will say now is just to recap. I think you covered this, but I was saying, are you saying that we can be assessed in future pre-cancerously for the possibility of future cancer likelihood, and then have an saRNA vaccine already prepared and stored away, maybe years in advance of it actually being required?
Um, Certainly we know that there are genetic traits that make you more susceptible to certain cancers. So it may be possible in the future to say, if you are at particular high risk, there is a vaccine that could be given to reduce that risk.
Yeah.
But in order to get there, we would need to see that those vaccines were effective against the cancers once they had been identified, to
Mhm.
know whether they might have a role at prevention. Because doing a prevention trial would be very large, very long-ended, and quite challenging to raise the money for.
Yes, yes, I see. Then we have a question from Stephen Room, who says or asks, how does viral load affect the probability of infection?
Viral load obviously does, for most infections, will increase the probability of infection. So the higher the viral load, the higher the amount of virus that you're exposed to, the more likely you are to become infected. And the same would be true for bacterial infections. The higher the bacterial load, the higher the chances of infection.
And then next question is from Linda White. Can this technology be modified to remove senescent cells? Wow.
I mean, the answer is the short answer is I don't know. The longer answer is there is a possibility that that may be something that can be done. The challenge there more is about whether, well, there are many challenges, but if you had an approach that could wake those cells up, is how efficiently can you reach all of the cells that need to be woken up? So it gives us the tools to start to explore some of those questions, but we don't have the answers today.
Thank you. And then finally, we have a question from Donal, who asks, is there any reason to think mRNA vaccines would not have wider effects, such as their potential preventive effect in Alzheimer's, as shown with more traditional vaccines?
So there are many. I mean, I've shown the positive things about RNA vaccines. There are many things that we still need to work on, and they're not going to replace all vaccine technologies. So we touched on, and I think even one of the questioners said, well, the durability of protection is not as long as we like. And it's certainly not as good or as long as some of the childhood vaccines. So measles, mumps, rubella, you're not going to see those replaced by mRNA technology. They are very good for pandemics because they're very fast. They give you a very rapid immune response. We do think that we'll be able to extend that immune response to, a number of years. It's unlikely we'll be able to extend it to a lifetime. But when I talk about that, that's about maintaining a level of immunity that's required to prevent infection. They will induce a memory response that does live for a prolonged period of time. So if you were exposed to something later, you'd still be better off than not having a vaccine. And in that context, for example, the vaccines that we're developing against Ebola. Ebola, untreated, has a mortality rate of about 50%. Now, you only need to tip the immune system a little bit. You still may be very, very sick from Ebola, but if you've been pre-exposed and you have an immune response and it's a memory immune response, it will probably be enough to prevent you dying from that infection for many years to come, even though you may be quite unwell.
I see. Thank you. That brings us to the end of our questions. And thank you very much for that, Robin. I'll hand over to David Southwood for a final word, please, David.
Right. Well, Robin, what a wonderful show. I was really very gripped by your presentation. And I was also, congratulations to the audience on some very good questions, I think.
Yes.
I mean, when some of our stars are over in Italy at the moment, it's a little worrying. I was worried that but I really thought you've taken us with you. And I'm really delighted we've been able to show how far we've come with you, Robin. It's a sign of really a very good show, but also something that's of profound interest and importance to us. Otherwise, we wouldn't have concentrated so hard, I assure you, on a Friday afternoon. So really, I would like to ask everybody if you can all unmute now. We can make a noise, or if you can find the symbol. Basically, this is a vote of thanks to Robin Shattock for really starting our weekend in a wonderful way.